Nature of genetic material. Actually, how the genetic material might be. So previously, scientists were thinking what might be the exact genetic material, whether carbohydrate, protein, or fat, or DNA, RNA. So to find out, scientists worked on this concept and they tried to focus towards the thing that genetic material is the DNA and DNA only. So prior to go towards the biological evidences to prove DNA as a genetic material, we will see some of the characteristic features of genetic material. We know genetic material is a substance which not only control the inheritance of the traits or characters from one generation to next generation, but also able to express its effect through the formation and function of characters. So this genetic material bears some of the characteristic features. One, storage of genetic information. So genetic material, no doubt, it is responsible for storing, it has the ability to store all genetic information about structure, function, development and reproduction of all living cells of the organism. Another characteristic is its accurate replication process. This genetic material replicates accurately and without any mistake, and that's why the same genetic information is transferred to the daughter cells or even in the next generation. Next characteristic feature is its transcription. So genetic material has power of accurate transcription, so that stored genetic information are transmitted to cell as and when needed. Chemical stability is another characteristic feature. So genetic material must have physical and chemical stability so that the stored informations are not lost, and that is there to this genetic material. Variation is the another characteristic feature. Genetic material should be capable of undergoing modifications by or due to mutations and recombinations, so that it can contribute to the variation and adaptations leading to the evolution of living organism. Now we will go towards the evidences which will prove that DNA is a genetic material. So there are some direct evidences and also indirect evidences to prove this fact. So first we will study related with direct evidences. So these direct evidences which proves that genetic material is DNA are these experiments are carried with microorganisms, and we can name them as due to the study of transformation of bacteria, then bacteriophage infection, bacterial conjugation and transduction. So transformation, transduction and conjugation, these three phenomena we will study, and also bacteriophage infection. So out of these four direct evidences, first one, that is transformation of bacteria. So scientist named as Griffith, see here in this figure, scientist named as Frederick Griffith in 1928, he discovered a phenomenon in bacterium named as a Diplococcus pneumoniae, and that phenomenon is known as a genetic transformation. So transfer of DNA fragments from dead bacterial cell to living bacterial cell, resulting in new genetic recombination, and this is called as a genetic transformation. So Griffith worked on Diplococcus pneumoniae and found two strains of Diplococcus pneumoniae, one wild strain and another mutant strain. In wild strain, bacterial cells are smooth in appearance because of carbohydrate capsule and are virulent, that is pathogenic, and are responsible for causing pneumonia. So this was the characteristic feature of wild strain. While mutant strain, bacterial cells are rough and non-capsulated and avirulent, that is non-pathogenic, while wild strain was capsulated. Here mutant strain is non-capsulated, and thus they are non-pathogenic and thus unable to cause pneumonia disease. So we have seen the characteristic features of these two strains of Diplococcus pneumoniae. Griffith done one experiment in which he injected non-capsulated avirulent strain of Diplococcus pneumoniae into body of the mice. For the first time, what he did, non-capsulated avirulent strain of Diplococcus pneumoniae, see here in this figure, he injected with the help of injection to the mice. So here bacterial cells were unable to cause pneumonia and the mice remained healthy. See here, mice didn't die. But when he injected capsulated virulent strain in mice, see here, capsulated virulent strain in mice he injected, and he came to know that bacterial cells cause pneumonia to the mice and mice die. Then next what he did, when he injected heat-killed virulent strain of Diplococcus pneumoniae, heat-killed virulent strain into mice, the bacterial cells didn't cause pneumonia, and thus mice remains healthy. When he injected mixture of, now this is the case C, actually here The strain was virulent but unable to cause disease and mice remain healthy. Now what he did in the last part of the experiment, he injected mixture of a virulent strain and heat killed virulent strain. A virulent strain, this one, and heat killed virulent strain, this one, into mice. And what he observed, mice were death here. And this is due to the pneumonia. So after post-mortem of the dead mice, it was found that mice were killed because of virulent bacteria. And from this result, Griffith concluded that presence of heat-killed virulent strain of bacterial cells might be responsible for the transformation of virulent bacteria into virulent bacteria. This phenomenon of transformation is also known as Griffith's effect, and more popularly it is considered as a bacterial transformation. So here we can see that the cause of a virulent bacterial transformation into virulent bacteria is the transfer of DNA. Again, later, Avery, MacLeod and McCarthy, these three scientists, they identified that the transforming principle or substance was DNA and not any other. So this is one proof, direct evidence, bacterial transformation. Now the another one, that is bacteriophage infection. So this is bacteriophage. In this figure you can see here the bacteriophage, and this is a bacterial cell. So bacteriophage, that is the virus that settles on the wall of the bacterial cell and sends its genetic material into the bacterial cell, that is DNA, into the bacterial cell. So by using bacteriophages, Hershey, H-E-R-S-H-E-Y, Hershey and Chase, C-H-A-S-C, by using radioactive tracer technique, they provided direct proof that DNA is the genetic material. The viruses infecting the bacterial cells are called as a bacteriophages. Viruses which infect bacterial cells are called bacteriophages. So T2 bacteriophage can attack and infect the bacterium Escherichia coli. So see here in this figure the transfer of this genetic material. This is the genetic material, DNA. It comes here. See, it is shown in this bacterial cell. It multiplies over here. Bacteriophages are produced 1, 2, 3, or like that many. And breaking of cell wall of bacterial cell, and that is called as a lysis. And these bacteriophages, they come out. See, this is the pattern. See here how the bacteriophages are produced inside the bacterial cell. Now what he did, Hershey and Chase, these two scientists, what they did, so they labeled they used radioactive phosphorus. They used radioactive phosphorus instead of simple phosphorus in T2 phases. And observed that whether this radioactivity goes into the next progeny, and they found that radioactive daughter bacteriophages are given out from the bacterial cell by the process of lysis. And this proves this is the another direct evidence that DNA is the genetic material. Now, third direct evidence is bacterial conjugation. Bacterial conjugation. See, conjugation is the process where two bacterial cells, one positive strain and second negative strain bacterial strains, they form a bridge in between and called as a conjugation bridge. And through this conjugation bridge, the transfer of genetic material, so that concept we will see here, bacterial conjugation, which will give you the direct proof that plasmid present in the F plus cell. See here, this is the plasmid. Plasmid present in the F plus cell. So it is not there in the F minus cell or female cell. But through conjugation bridge, this plasmid gets its entry into the F minus cell, and F minus cell now gets converted into F plus cell. So this experiment here in this diagram, we have seen how it happens. So bacterial conjugation can also give the direct evidence that the genetic material is a DNA, which converts F minus cell into the F plus cell. Then the another direct evidence, and that is transduction. Actually, transduction is a process of transfer of genetic material from one bacterial cell to another bacterial cell via viral agent, that is bacteriophage. Transduction is the process of transfer of genetic material, that is DNA, from one bacterial cell to another bacterial cell via viral agent, and that is the bacteriophage. So in this figure here, this bacteriophage is shown. So bacteriophage, it transfers the genetic material, its DNA, See, it is its DNA. It transfers this DNA to the another cell, from one bacterial cell to the another bacterial cell, and here this agent, viral agent, is required. So here just see how it happens. This genetic material DNA will come into the first bacterial cell. Then this DNA is here at present. Then this DNA multiplies over here, and this DNA becomes a part of host DNA here. And the bacteriophages, they are produced here. They also get the viral DNA or DNA of virus over here. Now this viral DNA, the same bacteriophage will come here and will settle on the another bacterial cell, and in doing so it will transfer its this type of DNA into the host DNA of this bacterial cell, which will become the part of this DNA of this bacterial cell, and later thus from one bacterial cell the transfer of genetic material to another bacterial cell takes place via this vector, that is bacteriophage. So these are some of the direct evidences. Now some indirect evidences. One of that is localization. All the cells of living organisms contain DNA, particularly in chromosomes. Reaction with Felgenstein have shown that DNA is entirely restricted to chromosomes, and DNA is the major component of chromosomes. This DNA plays a vital role as genetic material. Another indirect evidence, amount of DNA. So amount of DNA in different cells depend on number of chromosomes and sets of chromosomes present in that organism. The body cells or somatic cells have diploid number of chromosomes. The sex cells have, or gametes have, haploid number of chromosomes. Stability is the third indirect evidence. Studies on chromosomes reveals that genes are arranged in linear fashion in all the chromosomes. Particular length of DNA molecule constitutes a gene. Genes are stable. Gene or DNA replicate without any mistake during cell division, showing their stability. Rarely genes change, causing mutations in organism. So these are some of the indirect evidences. X-ray diffraction studies of Wilkins. Variety of X-ray techniques and methods are used in research work. X-ray diffraction technique is based on scattering of X-ray by crystals made up of different atoms. By X-ray diffraction methods, one can identify crystal structure of various solid compounds. When crystals of a molecule are exposed to X-ray, the X-rays are scattered according to the structure of molecule. Pattern of scattering of X-rays is unique to each type of molecule. These X-ray diffraction methods are extremely important as compared to X-ray absorption methods and X-ray fluorescence methods. In X-ray diffraction technique, a beam of parallel X-rays is exposed or directed on regular repeating structure of atoms. The X-ray beam is scattered by crystals and it gives a specific arrangement of atoms. Specific arrangement of atoms of different molecules. Diffracted rays are recorded on photographic plate. This is that photographic plate. And by analyzing photograph, one can formulate the structure of molecule. So with this introduction, introductory part, we'll go towards how the study is done. In year 1938, Asbury and F.O. Bell for the first time used X-ray diffraction method to find out DNA structure. Where they subject DNA fibers to X-ray diffraction analysis. By their X-ray crystallographic studies of DNA molecule, they concluded that DNA has high density, it is polynucleotide. Each of nucleotide was oriented perpendicular to the long axis of DNA molecule. Distance between two successive nucleotides is 3.4 angstrom. But drawback here of their study was the sample of DNA used by these two scientists, Asbury and Bell, were impure. And then later, during 1950 to 1953, Wilkins and his associates used highly purified DNA fibers to obtain X-ray diffraction photograph of DNA fiber. And his female associate, Rosalind Franklin, obtained superior X-ray diffraction photograph of DNA So this is related with the on what they observed, the salient features we can say due to their X-ray diffraction study technique. Wilkin and his associates, they concluded that three-dimensional structure of DNA for the first time in 1951 they tried or they have established. Also they concluded that DNA molecule is double-stranded and not linear but two strands of DNA which are twisted around each other to form a double helical structure. Means we can say that DNA consists of two polynucleotide chains wound around each other. One turn in each DNA strand occurs at 34 angstrom distance. That is also their conclusion. 34 angstrom distance of turn of strands includes 10 base pairs. It means 3.4 angstrom distance between two base pairs. DNA is organized in highly ordered or sequential helical form is the another conclusion. The DNA from several different species has identical spatial orientation. That is, DNA in different species has identical organization in specific space. So, by using the knowledge of this study later in 1953, James Watson and Francis Crick were able to give the model of DNA and which is world known.